welcome back to the new video. And in this video I'm going to describe today about a biotechnology process, that is the gene cloning. Okay, so this is one of the most requested topic. One of my subscriber, he wants, and some of my students, they also wants to learn this about this gene cloning. Okay, so from the name itself it is clear that it's a process to clone a gene, okay, a desired gene. And what is cloning? Cloning is production of identical copies, okay, numerous identical copies. So let's start. This technology, gene cloning, is also known as recombinant DNA technology. Okay, so nowadays if we talk about the molecular biology, then this is one of the most powerful technique of the molecular biology. Okay, so in this process what we do, in short, first of all we isolate. Sometime in the examination this only definition is asked, so just to give you a brief introduction, it's the process where we isolate a gene, okay, and then makes its multiple identical copy. Okay, so making of the multiple identical copy of a gene, okay, is the literal meaning of gene cloning. You understand? Okay, so there are basically 7 steps in this process. We are going to talk about one by one. Okay, I will cover this in very detailed manner so that you understand everything. So first step is, of course, isolation of target DNA or gene. Okay, so this is the first step. First you have to identify like which genes you want to isolate. Okay, suppose for example there is a gene which is responsible for the stress tolerance. Okay, that gene is called, suppose, A gene. Okay, so this gene is responsible for stress response in plants. Okay, and now just assume that this A gene is present in mustard plant. Okay, so but I am developing like I have a pea plant. Okay, or maybe, okay, let's say I have a tomato plant. Okay, so I will take the plants. They are not they are very weak to the stress. They do not perform well under the stress condition. So what I want is, I want to transfer this gene of the mustard to the tomato, so that my plants of tomato, they have the stress tolerance. Okay, and this stress tolerance in mustard is observed in the mustard. Why? Because of presence of this gene. Okay, so first of all, in the process of gene cloning, isolate the gene of interest. Okay, first select which gene you want to clone, and then go for its isolation. Now there are 3 ways, okay, for gene isolation. First is PCR amplification. Second is restriction enzyme. I will write it in short. This is restriction enzyme digestion. And the third is the chemical synthesis. Okay, so let's talk about this PCR amplification. If I know the sequence of this A gene which we are talking about, okay, if I know the sequence, what I will do, suppose this is the starting, okay, of the gene, and it ends with the stop codon. Okay, now there are other nucleotides also. If I know the specific sequence, okay, and flanking sequence also, okay, if I know the nucleotide sequence of this gene, what I will do, I will design a primer, okay, that are specific to the gene. Okay, primer I will design, I will write here gene specific primer. Okay, so whichever gene we want to clone, we will draw the gene specific primer from it, and with the use of the PCR amplification, we will amplify those or multiply the copies of that gene. Okay, so how the PCR is used to clone a gene, I have discussed in one of the another video. I will post the link into this video also in the description box. Okay, so that you can go and watch it there. So if the gene sequence is known, we will go for the PCR amplification. So the next method is the restriction enzyme digestion. Okay, if the our gene of interest is located within a DNA fragment that has the restriction site flanking to it, then we will use the restriction enzyme to cut the DNA. Okay, so this is the DNA, suppose. Okay, so DNA is this is a stretch of DNA. Okay, so now we have here restriction enzyme sites. Okay, restriction enzyme sites, and our restriction enzyme sites. Okay, and our gene of interest is suppose in this region. Okay, so what I will do, I will just cut these sides. I will just cut the DNA segment using restriction enzyme. Okay, so that I will just have this DNA segment, okay, that contain the gene of my interest. Okay, so before moving on to the chemical synthesis, I hope everybody know what is restriction enzyme. Restriction enzymes, they are the enzyme that have the potential to cut the DNA at a specific recognition site. Okay, so then we'll go to the chemical synthesis. We'll go for the chemical synthesis of the gene if the size of the gene is small. Okay, because it's a costly process, and in this process we will chemically build the gene sequence nucleotide by nucleotide using the automatic DNA synthesizers. Okay, so there are DNA synthesizers that will help in building the gene sequence. Okay, so this is all about the first step, that is the isolation of target DNA or gene. Okay, so the second step is the vector preparation. Okay, so we know that vector is a DNA molecule that can carry the gene of interest into host cell and replicate. Okay, so what are the duties of vector? Okay, it will take our gene into host cell. Okay, and why for the host cell? To replicate it. Okay, okay, so there are some common vectors like which we are regularly using in the labs, that is the plasmids. Okay, then there are cosmids also, then bacteriophages also. They served as an excellent vector, okay, in the scientific studies. So these are the common vectors which are used in the labs. Okay, so I told you in the first step that we will cut if we are using the restriction enzyme cutting of the DNA, then we will if we are going for the restriction enzyme digestion of DNA segment, then what we will do, we will cut our vector also. So with the restriction enzyme. Okay, so the vector also, they have the restriction enzyme sites. Okay, and why we are cutting this? Okay, because now we have a DNA segment. Okay, this is a gene or DNA segment of our interest, and it has the cuts, it is cut by the restriction enzyme. Okay, now we have to introduce this into the vector. Okay, so I will just draw vector. Suppose vector has the restriction sites here. Okay, so what we will do, we will digest the vector also. This is vector. Okay, so what we will do, we will digest the vector with the restriction enzyme. Okay, okay, so after digestion the restriction enzyme will cut this DNA fragment here. Okay, and it will generate the sticky ends. We will choose the restriction enzyme in a way so that it will produce the sticky end. And why these sticky ends are needed? Because our this segment has to go here, it has to stick to this portion of vector. Okay, and to do so we will cut the vector with the restriction enzyme. Okay, only if it has the sticky end, then only this segment will go it here. Okay, it will act as the sticky end, it will act as a glue for sticking the our gene of interest into the vector. Okay, so now this is vector. Okay, uh, which is uncut. If we cut the if we digest the vector also, and then this our DNA segment has attached there, now it is called as recombinant DNA. Okay, so if it has a gene and this is our vector body, then now this is called recombinant. Okay, because a new recombination has taken place here. Okay, I hope this is clear. So I explain you that we will go for the ligation of this linearized DNA into this vector. Okay, and when we are doing this process is known as ligation. Okay, so for the ligation we have a specific enzyme that are known as DNA ligases. These enzymes are responsible for joining the DNA fragment with the vector by forming the phosphodiester bond. Okay, so the complementary sticky ends of the target DNA and the vector, I already explained you, they will annihilate each other, and the DNA ligase, this will seal the leaks. Okay, seal the leaks. Okay, by forming the phosphodiester bond between the two. Okay, so this is the ligation process. Then comes the step 4, that is the transformation. Okay, so guys, I will repeat it again. In the first step we have selected our gene of interest, we have isolated it. Okay, and in the second step we prepared the vectors, and then we did the joining of both vector as well as the gene, and that process is known as ligation. Now we have ligated our product. Now this recombinant molecule, DNA molecule, it has to be introduced into the host cell, which is typically bacteria. Okay, so when this R DNA, usually we are calling R stand for the recombinant. Okay, if you write rDNA also, it is fine. So this recombinant DNA, if it is transferred to the host cell, okay, which is usually very bacteria, because it is very quite easy to work with the bacterial system. Okay, so when we transfer it here, I will keep it like this so that it is easily visible to you. Okay, then this process of transferring it into the host cell is known as transformation. Okay, so there are basically two popular methods for doing the transformation. First is the heat shock, and the another one is the electroporation. So one thing I will tell you guys, I really like biology in the sense that most of the time from the name itself it is clear that what we are going to do. So in the heat shock method also, what we are doing, we are giving the bacteria a kind of heat shock. Okay, so that heat shock will create pores in the cell membrane, and it will allow the DNA to enter inside the bacterial cell. Okay, and the electroporation also, it's like we are providing the electric pulse. Okay, high voltage electric pulse, and this high voltage electric pulse, it will do, it will again then create the temporary pores in the cell membrane, and the DNA, it will enter into the host cell, that is the bacterial cell. Okay, so now we have transformed our bacterial cell also. Okay, so and we have the replication has occurred. Now how to select, it's not possible that all the bacteria will be transformed, right? Then we have to, the next process or the next step in the gene cloning is we have to select that which bacteria are transformed and which are not. So how we are doing, we are putting the bacteria generally we are growing in the media. Okay, so to select the transformed, we will put a selective media. Okay, now here why we are using the selective media, this is the next question. So our vector body, okay, it has antibiotic resistant site. Okay, and it has origin of replication also. Okay. So those bacteria which were actually transformed, they will, because the vector backbone, it has the antibiotic resistant side, then they are resistant to a certain antibiotics. So we will put those antibiotic into the media, and we will select only those cells, like only those cells will survive which will have the vector backbone. Okay. So suppose this is a vector. Now this vector has no gene. Okay. This is our vector, and blue one I will join. Okay. So this is our recombinant DNA. Okay. So now what will happen? Sometime the vector, it will join itself only. Okay. And recombinant DNA, of course. Now both this structure, they will have the antibiotic resistant cells. So sometime what happened that even this these colonies will survive. Okay. The untransformed bacterial cell will die. I will write the untransformed bacterial cell will die. Okay. So once we have done the transformation, we will have three different kinds of the cell into the media. So what kind of cell? First of all, the cells, we have the vector only, then there are few recombinant DNA, okay, and then there are untransformed cells. So when we are plating them onto the selecting media, this untransformed bacteria, they will die because they do not have any antibiotic resistance in them. However, okay, and these two will survive. Okay, why? Because the vector itself have the backbone, okay, and the backbone contain the antibiotic resistance. So we will go for the screening, okay, of the cells. So there are numerous methods for the screening of the cell. I will write three. First is the colony hybridization. So if we know the, like, we can design the primers that are specific to our gene of interest, and then we can do the hybridization reaction, okay, and amplify, and we can find that our colony is our gene is present in that colony or not. Okay. So what are colonies? I will tell you. This is our plate that has the nutrient media. Okay. So the bacteria, it will grow like small round structures. Okay. So this is known as colony. This is one colony. These are all different colonies. Okay. So we will do the colony hybridization. Then we can do the PCR screening also. Okay. And we can go for the DNA sequencing also, okay, to confirm that the gene is present there or not in the transformed cells. Okay. Then the last step is the amplification. Okay. Once we have the after the screening process, now we have the positive colonies. The positive colonies are the one that contain the recombinant DNAs. Okay. So then we will produce them into large culture. Okay. Okay, for the protein expression, for the various application. Okay, the first is the protein expression, or for the gene therapy. Okay, so these are the 7 steps into the for the gene cloning, friends. So now quickly talk I will just in the 4th step I will give you the application of the gene cloning. Okay, gene cloning. So why do we use the gene cloning? We use gene cloning for the production of recombinant protein. Okay. Okay. It is used to produce large amount of therapeutic proteins such as insulin or the growth hormone. Okay. Then I told you here only that we can use the gene cloning for the gene therapy purpose. Okay. The third one we are discussing already in the video, that we can use the gene cloning for the genetic engineering of okay, plants and animal. So if there is a desirable trait in a gene like stress resistance or like immunity boosting tendency, something like this. Whatever is your interest, based on that we can use the gene cloning, okay, and transform the species, okay. And the last but not the least, the basic research is sometimes, we don't know the function of the gene only, okay. So just to find out the gene function and to characterize it, we will do the gene cloning process, okay. So I hope this is very clear from this video that it's a very, very powerful technique. I will write it here. It's a very, very powerful technique. And now if we talk about the molecular biology, we cannot just think the existence of the molecular biology without PCR and this gene cloning. And this process has revolutionized the molecular biologist, okay, and it has opened a wide range of possibilities for research and application. I hope it is clear to you. Thank you for watching the video.